Amit, please.
Very good morning. Thank you, Dr. Shivaji, for the kind introduction. Also like to thank Dr. Thangraj and Dr. Keshav Singh for having me over here. As what already mentioned, I will be talking about a slightly different aspect of the diversity and health disparity. I will be talking about the somatic aspect of it, and that is associated with the cancer. So I start with a brief introduction by putting things into the perspective that how the landscape of cancer incidence, I mean, Dr. Lachance has already kind of like alluded to this fact earlier in the earlier session. So this is how the burden of cancer is projected to change over the next few years as the data that comes from the GloboCan. What is so surprising and what is so remarkable over here is that the majority of these burden are going to be contributed by the countries with a middle or lower income group, that is countries like China and India. As what you see that the countries with a higher income group, there is more or less kind of like a plateau. In fact, now I would show you like in a couple of slides down that the disparity is kind of like increasing, that this is not just a plateau. With the recent reports, we already kind of like get to see that there is already a kind of a trend of a decline as well. But this unfortunately doesn't happen to the case when it happens to be the countries with a middle or lower income group, such as in India. So Dr. Lachance has already kind of like alluded to this. I wanted to walk you through this, that how the landscape of cancer in India is distinct than what you get to see in the countries in the Western world. So, for example, the cancers such as pancreatic cancer or melanomas or prostate cancer, which are one of the major cancers in the Western countries, that do not figure in the major cancers here in this part of the world. What we get to see, something which is kind of like of a higher predominance, happens to be, as what Dr. Lachance has already mentioned, is that the cancer of the head and neck, the lip and the oral. If you would combine at all these sites, this is where majority of the cancer among men, that what you kind of like get to see. And when you come to the women, again, the cervical cancer, most of these cancers, the cervical cancer and the head and neck cancer, these are not like the major cancers in the western part. So now, if you're wanting to study these tumor types, the initiative to these has to happen around from this part. And so my laboratory has been emphasizing on to understanding the underlying genetic alterations and to emphasizing on to the diversity as what exists out there in the Western world, and if these treatments or whatever has been kind of like been carried out, if it could be directly something that could be adopted here in the Indian settings. So I will give you two different examples in my talk, emphasizing onto this diversity and how it could be kind of like implemented here in the Indian settings. In particular, I'll be talking about lung cancer, initially from my lab, and I will also be talking about gallbladder cancer, something which is very much considered to be like more predominant in the Indian region and the South American, but otherwise in the rest part of the world, it is seen as a rare disease. So what is also kind of like really underlines these diversity is what kind of like comes out from here is that the cancer incidence in India over the period of 1990 to 2016, somewhere from 0.5 million, it has changed up to like 1 million. The incidence has kind of like gone higher. What is more interesting is that that the cancer deaths which are associated over here, that is kind of like significantly high, from 0.3 million to 0.8 million. So definitely there is a huge void in understanding the landscape of understanding these diseases in the Indian settings, and that's what my laboratory is primarily focused on. This is an old slide, but kind of like emphasizes the difference that between the two different worlds and the cancer and the other different diseases. So this is intentionally kept on to be like old. This is an old slide from 2001, and here is a snapshot between 1950 to 2001 across different diseases, whether it is cardiovascular diseases or cerebral vascular or pneumonia or infectious diseases. What you get to see is that the drop in the mortality in these diseases have been significant. However, when it comes down to cancer, you do not see this significant or any drop per se. Now, if you would slide this window to 2020, this scenario hasn't really kind of like much changed. In now that's where the disparity kind of like comes in. If you would see this in the Western world, this is what kind of like, this was a report from last year, 2019, and in the recent report kind of like talks that 2% furthermore decline in the deaths. So basically it kind of come down to 27%. Now this is something which is very much restricted onto the Western world. This does not kind of like, this is far to even imagine in the countries like here in India. So there is certainly, we have been deliberating of like the diversities and based on the genetic aspects, but there are several other different aspects that are kind of like also associated around with. So what I'm going to talk about today, I'm going to talk about is two different major cancers, two different Indian specific major cancers in that case. One is like lung cancer, which is across the globe one of the most common cancer found in men, and that also happens to be as a single site in India. So this work has been done in collaboration with a medical oncologist friend of mine, Kumar Prabhash, and my first graduate student, Pratik Chandrani, that what we kind of like talk about. This is a textbook example now which has gone into the textbooks, something similar to what you may have like read in your textbooks of like the Philadelphia chromosomes, this in CML, where Gleevec has been the wonder story. Here is a patient of a CT scan of a patient of lung cancer, and here what you see are the nodules, and this patient happens to be positive or harboring a mutation in a tyrosine kinase, that is an EGFR. And this patient receives an EGFR inhibitor not through infusions, but receives orally a single inhibitor, and what you get to see is that in the period of 2 months, the complete remission. So this is the kind of like a proof of principle. What follows, whether the resistance develops or not, that's a separate question. The question is, the point over here is that should these kind of like alterations could be identified, this is the kind of like dramatic response that you kind of like get to see. And off to mention about it, since this discovery in 2004 of the EGFR mutations in lung cancer, till 2004 around, if you would look at the survival of lung cancer patients, which was then about 7 to 8 months for the stage 4 disease, today if those are harboring any of these, several of these bunch of these therapeutically relevant alterations, it has gone up to somewhere around 60 months. So that has been the impact from 7 months to 60 months. So when I started my laboratory here in India, and that was the early of 2011, we were wanting to the first question that we wanted to ask is about like the profiling. We wanted to ask is that if EGFR mutations, which occurs in lung cancer, how does it vary by ethnicity? Because it has been known that EGFR mutations in the Caucasian population occurs at a frequency of somewhere around 10 to 15%, and in the East Asian population, it's somewhere from 35 and higher. We do not really know what is underlying genetic reason for it, that why is there a disparity. When it comes down to the Indian settings, we still did not know at that point of time that what is the underlying EGFR mutation frequency in the lung cancer patients of Indian origin. So we took one of the first studies with 1,000 odd patients at Tata Memorial, and we could establish that the EGFR mutations occurs at an intermediate frequency between the Caucasian population and the East Asian population at 23%, and since then it has been kind of like revalidated by several studies, and this has become like on the basis that the EGFR mutations occurs. Why it happens this way, we still really don't know. Whether it's an amalgamation of different populations, we really don't know. What we do see is that that those patients in the Indian settings is that those patients who are harboring EGFR mutation, when they are given orally EGFR inhibitor, a study done at Tata Memorial, Mumbai, they do respond, and they do kind of like do better when compared to those which are EGFR negative. So these were the kind of like embarking onto like profiling experiments. That's how I was kind of like establishing myself in the laboratory. I mean at ACTREC, Tata Memorial in the initial days, where we beyond EGFR we went on to do with the KRAS mutation profiling and others, and we could see that these kind of alterations that has been associated with these diseases, they do occur in the Indian population as what and you would be expected to be, but they're occurring at a different frequency and at a very significantly different frequency. While the EGFR mutations were happening at a higher frequency at 23%, mutations in lung cancer adenocarcinoma subtypes was found to be happening at a much lower frequency as compared. So we still do not know what exactly is the basis of it and of what is causing this diversity. Going deeper into the lung adenocarcinoma, we wanted to understand that beyond the EGFR and KRAS, are there other different genetic elements? Let's see how are they kind of like doing it in lung cancer. So this was one of the targeted sequencing based approach, where we took like a discovery set of 125 odd samples and a validation set of 400 odd samples. The validation was done by Svicanome, then and Agena now, mass spectrometry based, and this was based on the next generation sequencing. So I'll cut a long story short how exactly it was done. That really is not significant today. What we found was something kind of like interesting. What we found is that, as what I said, that the major players happens again to be like the similar. If EGFR mutations is known to be kind of like occurring in lung adenocarcinomas, we do find those similar mutations in the Indian lung adenocarcinoma patients as well. But what we did find is something more striking and interesting at that point of time, is that we found that a novel tyrosine kinase, that is an FGFR3, which has been shown not to occur in the TCGA data sets. I mean, several of the lung cancer sequences that has been performed in the Caucasian population, they did not find at this frequency of FGFR3. We found some novel mutations, and they were occurring at a very significant frequency of 5.5%. These mutations were plural. They were in the kinase domain, they were extracellular domain, and they were occurring in a total of about 5.5%, which was not reported in the Caucasian. Now, 5.5% of an occurrence in an Indian lung adenocarcinoma patient, given that we have a huge denominator, itself means that it would be a total burden of non-Hodgkin lymphoma in India. So that means this is a disease in its own settings. My laboratory, what it does is that it does not just catalog different alterations, but we also kind of like functionally validate we pair our discoveries with the validations, physiological relevance of the alterations that what we find. So here is a study in head and neck cancer, in oral cancer, and this patient was harboring an FGFR2 mutation in the same FGFR family, and this was shown to be an activating mutation. And similar to the lung cancer, as what I showed you earlier, this patient, when receives orally pazopanib and another tyrosine kinase inhibitor over a period of like 2 weeks, such is the kind of like a dramatic remission that you kind of like get to see. So the question that we wanted to ask is that if such could also be kind of like brought to the FGFR3 mutations that what we have kind of like found here in the Indian lung cancer patients. The first thing that you would want to do is like if these novel mutations that what we are finding, if they are kind of like physiologically relevant at all or not. So what we do is that we take it to the cells, we overexpress in isogenic system. What we find is that indeed that these cells were causing the cells to get transformed. And these cells which were transformed, they were found to be sensitive to the FGFR inhibitors, which were commercially available. We went on to show that those cells which were overexpressing these mutations that were derived from the patients, they were constitutively activating the pathway, and they were sensitive. These pathways were abrogated when you add the inhibitor in presence of the inhibitor. We went on to go ahead and like validate onto the xenograft assays into the mice as well, and then go back to the patients and all kind of like do the validations. So basically what we find is some novel discoveries in FGFR2, and what do we do from here on? Starting a new clinical trial. Now that's where the major impediment kind of like really kind of like happens out. We have still not been able to kind of like initiate a clinical trial with the FGFR3 for several reasons, and one of the primary reason is that we really do not have the salt. We do not have the inhibitor here available in India, such as pazopanib, that can be used in the clinical settings on compassionate ground perhaps. I mean, while right now I'm talking to you, we have at least more than 20 or perhaps more than that of a patient who are stage 4 lung adenocarcinoma disease patients who are harboring FGFR3 mutations. So the hope is, if this discovery from the basic science, from the basic laboratory, could actually be translated onto the clinics. We really don't know as of now. But that's what the kind of like the hope is. The second story that I wanted to talk about today, switching gears, is on gallbladder cancer. That here This is something in collaboration with a surgeon friend of mine, Shailesh Shrikante, and another graduate student, Prajesh Iyer. So gallbladder cancer is something unique in its own settings. Like if you would look at its distribution across the globe, you will find primarily in the South American region a bit of it, and then a huge burden in the Indian subcontinent, and primarily even in India you find the disparity that something around the northern part by the Ganges, the incidence of gallbladder cancer is very high. However, when you move around towards the south, then you do not find that kind of like a higher incidence of gallbladder cancer. What exactly is the underlying reason? We really do not know. What we do know is that a majority of these gallbladder patients, they do have gallstones. However, the other way it's not true. If you would see that all the patients, all the individuals who have gallstones, less than 2% of them, they really kind of like go on like to develop gallbladder cancer. So there has been, gallstones have not been shown to be one of the predisposing kind of like factors. So we wanted to kind of like, the only predisposing factor that as of now that is kind of statistically significant that comes out in gallbladder cancer is being women. So this gallbladder cancer, we know that it's higher among women as compared to like males. But before I get into the gallbladder cancer, that what exactly we did, I would also want to talk about, just a detour one slide, I want to talk about that there was something else which kind of like gets into the story, is that my laboratory's long-time interest is also to look for the pathogens which are associated with cancer. We do like computationally subtract and then kind of like look for novel pathogens which are associated with the human tumors. We have developed several tools of that, and we have been analyzing the several of the TCGA data sets in the clouds. So one of that story that we kind of like published earlier, where we had developed a tool based on computational subtraction platform, which is an Hpv detector. It is freely available for download. More than 250 labs are using, and with like a lot of like reviews already been published about Hpv Detector. What it does is that it gives you a binary answer, whether any kind of data set that you have, whether it is an exome, whether it is whole genome, transcriptome, targeted, or whatever, GUI based for biologists, it tells you whether Hpv is present there or not, and at the same time it also tells you where exactly it is integrated. So I'll tell you why I'm talking about this, because this was something that was used in gallbladder cancer. And the other thing is that before, the earlier story in the lung cancer, what I talked about was the targeted sequencing, right? So, but we wanted to embark into something which was like a whole exome sequencing and whole genome sequencing. But before you want to kind of like do that, you would, and because we are into the somatic settings, we would want to get rid of all the confounding factors. The fact is that we do not yet have Indian germline SNP database. Those alleles which are occurring at like a lower frequency in 1000 genome or other, that we do not really know how exactly what is the effect of those, but we need to kind of like get rid of that, otherwise those will be confounding our somatic analysis. A lot of initiatives has been done since then. One of the early initiatives from my laboratory was developing a TMC SNPDB database, which was based on only a modest effort by 62 normal samples. Paid normal samples from cancer patients, and these normal samples we perform on whole exome sequencing, and we went on to go and find something which was about like 100,000 odd variants which were significantly there. But since then we have kind of like moved on. Now we have an in-house database of around 250 normal samples, and but now a lot of like other efforts have been kind of like done, such as Genome Asia or the indigen efforts, where this is going to get like much more, in which we are already benefited with the Genome Asia report to get rid of the confounding germline noise in the somatic data sets by using the Genome Asia and the TMC SNPDB. And we are hoping that what Dr. Vinod has already alluded to, the indigen is going to be like a major game changer in the field. So now, what we did with these, with these tools is something that I'm going to kind of like elaborate. This was like one slide. I wanted to introduce you that why exactly gallbladder cancer here that we wanted to kind of like study. I've already mentioned that the incidence is very high. In fact, the incidence in the northern part of region is much higher as compared to the other thing. We really do not know the major predisposing factors about it. So and given that the long-term interest of the gallbladder cancer of the laboratory, we were wanting to ask that what exactly kind of like leads to the gallbladder cancer occurrence, and so we were one of the early ones to kind of like to propose carcinogenesis and a dissemination model for the gallbladder cancer, something akin to something similar to what has already been done for colorectal cancer, the Vogestein model that you're already aware of. So what we found at that point, that means I'm 2 minutes or 4 minutes?
Yeah, 3 minutes.
3 minutes, yes. So what at that point of time they found is that that we found that the gallstones or the gallstones are kind of like associated with which have been known to be associated, that leads to a histological state as metaplasia, leading to different states, and this is how a sequential progression of the gallbladder cancer. Not going to the details of it. Pratisha here went on to kind of like, and what was interesting in this model was that we found that what was central to the progression of the gallbladder cancer was an inflammation. But we did not know at that point of time what could be the source of like that inflammation, because we know that the gallstones definitely are not significantly associated with the progression of the gallbladder cancer. So twisting the Hpv detector that I just kind of like introduced, what Prajesh did, he increased the reference database by using different pathogens, and what he found in an unbiased analysis that there was a strong association of salmonella, non-typhoidal salmonella with the gallbladder cancer. In fact, now since then there has been like a lot of like reports where people have used gallbladder organoid culture and they have challenged it with like salmonella strains, and which are already harboring MYC amplification and p53 mutation that leads to the transformation. So, but what Prajesh wanted to do after this this pathogen associated is that he wanted to go ahead with the whole exome sequencing and to look for different alterations. I will cut a long story short. We found that one of the significant alterations was in the ERB2. ERB2 is another tyrosine kinase which was kind of like found to be very significantly associated in gallbladder cancer among Indian patients, and the hope was that if ERB2 treatment, which is already kind of like approved in breast cancer, if that could be adopted for the gallbladder cancer. This was again an independent validation of the genomic analysis by the proteomic analysis. He took some cell lines and he did phosphoproteome profiling, and again he found similar that is the ERB2 to be the main culprit, ERB2 and EGFR co-amplification, and he could show that EGFR and ERB2 kind of like could be pulled down with they kind of like dimerize. So I will cut this story short. What he found, something very interesting in gallbladder cancer, is that besides the ERB2 that was significantly altered in the Indian gallbladder cancer patients, he found that there was another mutation that is in the KRAS, which is downstream to ERB2, and he found that different patients are harboring we found these patients to be harboring different alleles of KRAS. So they were harboring like this guy is harboring a wild type KRAS, which is harboring a mutant KRAS, but a different allele G41V, and here is a cell line which is harboring G12V. And if you would see that if we would knock down ERB2 in these guys, here there is this cell was found to be sensitive. I'll just take a minute to kind of like wind up. So this was sensitive, this was partially sensitive because this is a partially active allele, and this cell line was found not to be sensitive at all or not to be responsive at all to the EGFR inhibitor. So basically because KRAS is epistatic to the ERB2, and he could show that by the genetic ablation, and at the same time he could show that by the knockdown of the by the pharmacologically as well, by using treatment which are approved for breast cancer. He could show that the same thing into he takes the story all the way to the mice to confirm the story that the patients who are harboring ERB2 alterations in gallbladder cancer, we also need to look at the KRAS mutation allele, and these KRAS mutation allele will kind of like tell them whether these patients should be given as an anti-EGFR, that is an anti-ORB2 treatment or not, that kind of like comes out. So what model kind of like comes out, which kind of like gets translated now straight away from the lab to the clinics, is that here is a gallbladder, here is something which is already approved for colorectal cancer, and that we are kind of adopting to the gallbladder cancer. So what is happening is that here is a patient who is already harboring who's having only an ORB2 alteration. He's harboring a wild type KRAS allele. This patient gets to receive the anti-EGFR therapy. However, if there is a patient which kind of like harbors a mutant KRAS, as what we kind of like showed, they need to be precluded from the anti-EGFR therapy. That's what is kind of like now already kind of like being used at the Tata Memorial Center based on the genotyping that what we kind of like do. We have also got the NGS to the clinics, where we are kind of like generating different automated reports, but I would not take you into the details. Basically, we talked about in lung cancer about the EGFR and KRAS mutation profiling, and during this profiling, what has really come as an outcome is that something that today the profiling, every patient walking at the Tata Memorial Thoracic Unit, for them the genotyping is being done. It has been brought down the cost because of this analysis, is that from using non-proprietary reagent from $250 to about like $12, which is being given for all the patients. We talked about the FGFR3 alterations in lung cancer. I briefly mentioned about the anti-EGFR treatment that is going to be rolled out to the lung cancer, and in fact we have found like patients which are kind of like harboring ERB2 and KRAS in our NGS analysis there as well. Here is the lab. It's kind of like a big lab. People who are kind of like involved in the functional work. Here are the computational teams. We work on several different kind of like cancers. Here are the collaborators within the Tata Memorial Centers. Have not talked about most of like them. We're generously funded by the Tata Memorial Center, by the Department of Biotechnology, Wellcome Trust, DBT India Alliance, DBT VNCI, SERB DSD, and the Terry Fox Foundation. Should there be some time and interest, I'll be happy to take questions.